screen. Okay.
All right, we are now recording. And
so you should see an empty screen at this point.
Yep, we see an empty screen.
Okay. Now you should
now we see your slides.
Okay, excellent.
Thank you.
So thank you very much, Kate, for the invitation to join you today. I have attended a few of those meetings. I've enjoyed them, but also a little bit of time conflicts at times. So I've watched some of them offline. I am not really working on synthetic cells and things like this, but I'm going to be talking about data. I'm going to be talking about modeling, and I hope that this is going to be generic enough that you may find that useful. So I'm going to, I'm going to tell you a few words on how did I get into synthetic biology. And in the early 2000s, I was actually not in academia. I was a computational biologist working for DuPont. I was in Iowa in their subsidiary that does plant breeding, transgenic crops. And my job was to develop models of gene networks that could be used to guide breeding programs. And so I came across the first papers on artificial gene networks and got back to the leadership and said, look, this is really cool because this is predictive, all sorts of things that we can do in, with respect to that can change the way we're developing transgenic plants. And I actually started a synthetic biology projects at Pioneer, like in 2003, 2004. That's when I met Jim Collins. We licensed the toggle switch at that point to try to implement one in corn and that kind of thing. So I got into it fairly quickly. It was very early. It was very exciting for a year and a half because money was not an object. And then eventually DuPont didn't make its profit objective and they fired 25% of their research and development staff in 24 hours. And after that, it was a nuclear winter. So we couldn't do anything anymore. Everything was shelved. And so I looked for better opportunities. And that's how I got back into academia, spent 10 years at Virginia Tech, starting a synthetic biology group. The goal, the initial plan was really to develop, have a research group that would have both a computational and experimental activities. On the modeling side, we, most of what we did, the main project that we worked on was the development of a BioCAD tool that was called GenoCAD. We got NSF funding, and that was a web-based application. That was one of the first one, and I'm very proud of the fact that we actually managed to keep it running for about 10 years, which is good in terms of longevity of synthetic biology software. We got a number of papers. We got hundreds, if not thousands, of users. But the thing that was very frustrating is that the experimental validation of this software was very much lagging behind the software development. And I think it was a number of different reasons. One, the budget for experimental work was a little bit more limited than the budget for software development. I also found it somewhat difficult to start the experimental work. So, it was more difficult. I found it more difficult to find people, who understand both the software and the experimental part, and connecting the two seemed a little bit more difficult. I also had been at that point, out of a lab for probably the best part of 15 years, and I probably somewhat underestimated the difficulty of running an experimental program. And so that's pretty much the conclusion that I was reaching in 2016 when CSU asked me that I wanted to, move west. So at that point, I made a couple of decisions. One, I decided that my lab would focus on the experimental work, and that I would spin out all the software development to a company called GenoFab. The reason is that software is really important, but academia is not necessarily the best place to do software development. One of the reasons is that it's very expensive and you get very little academic credit for developing good software. Difficult to find people with software engineering experience in an academic environment. And also we wanted to broaden the scope of the software instead of just focusing on design, which is a very small section stage of a project life cycle. We wanted to pretty much like connect all the dots, all the stages of a synthetic biology project. And actually, I've also come to the conclusion that this whole idea of designing experiments, like implementing them, collecting data and analyzing it, it's not something that is really, that's something that synthetic biologists have really put forward, but this is something that can be used in more traditional, like basic biology research projects. And for instance, one of the projects that we worked on when I came here, when I came to CSU, I was just got an award from NIH to develop a stochastic model of the cell cycle, cell division in yeast. And that was a collaboration with my colleagues at Virginia Tech. And we can really, even though it's really more like systems biology and a basic biology project, it kind of raises a lot of the same issues as many synthetic biology projects. So the goal of this project was to validate and expand a model of the yeast cell cycle that had been developed over the last 20 years by John Tyson and his students. The latest version of this model had been published in 2015. It was a very large model. I mean, there were more than 200 state variables, close to 500 parameters. And the model was validated by showing that by changing the parameters according to the genotype of different mutants, you would have the same phenotypes in terms of whether the mutant was viable or not, or able to grow in different media. So the model had been developed using mutants that had been described in the literature, and they had been produced on using different background strains. The phenotyping was not, standardized, so there was a lot of interpretation that needed to be done, and in order to, compare different mutants. And sometimes it felt a little bit that, you were using different mutants that were a little bit like apple and oranges to fit one model. The other thing is that when you have so many parameters, like, when you have more parameters than mutants, you can start wondering if your problem is a little bit underdetermined or not. And so part of what we did was, okay, so instead of just, collecting, building the model based on previously published mutants, we decided to generate a large set of data that could be, that would be cleaner and that would be larger, so that, we can better validate the model and refine it. So we took 36 genes that were known to be involved in the regulation of cell division. Some of them were already in the model, others were not in the model. And we made the crosses 4 times using different sets of markers. And, I'm going to spare you a lot of details. But we made the crosses 4 times. So think of it as 4 replicates. all the crosses should, give more or less the same results. We also look, that yeast has two mating types, A and alpha. So we looked at both mating types in terms of the phenotyping. And so when you look at all the details of this experimental design, we could have potentially generated more than 10,000 strains. We only got about 60% of them representing 630 double mutants. And then we tested them in 6 different growth media, and depending on the carbon source, the cells grow in different at different rates. We were also adding some mutagenic agents, chemical agents that trigger some checkpoints. And so 6 different mutants, give you like different insights on the cell cycle. And we evaluated the phenotype by measuring the growth rate by looking at colony size at different time points. So the workflow kind of looked like this. There were some crosses producing heterozygous diploids, and we selected the double mutants, and then we produced some plates to do the phenotyping. So the point that I mostly want to make is that the raw data were like, colony sizes on those plates like this, but the experiment was pretty big. I mean, that's a small fraction of the number of plates that we have generated. So, when you have so many plates, it raises all sorts of issues in terms of not getting lost in the process. It was somewhat of a surprise. We ended up, the reason why we did the crosses 4 times is that we noticed that the results that we were doing when we did the first crosses were not exactly what we were expecting. And when we did the crosses multiple times, and when we look at the phenotypes of the mutants on the 2 different mating types, we were not always getting the same results. And there are some technical reasons for that, some of the limitation of the assays. But this was a little bit troubling. So we ended up with, having to reconcile a lot of replicates of those experiments and try to gauge the confidence that we had in those results in the triangle that you see on the side, and also compare our own findings with what was published in the literature previously. And it turned out to be much more complicated, than we anticipated initially. It was certainly much more complicated than the way the project had started, by just assuming, a single phenotype per mutant to fit the model. And so that raised a number of different questions. And we did something similar with the growth rates in different media, and I'm not showing that because it's not, it's not a seminar, but it's the cell cycle in yeast. So what is important is that we learn a number of lessons. I certainly learned a number of lessons in this experiment, in this project. One is that it's extremely challenging to manage a high throughput experiment like this. And I was aware that, data needed to be managed in the right way. I probably made the mistake of trusting too much people telling me that they knew what they were doing. Essentially, we spent about 2 years, a little over 2 years, running the experiment the first time, only to realize that, we were producing data, but I was not seeing a whole lot of results. And eventually, I decided to just, prevent people from getting into the lab until we analyze the data that they had already collected. And what we saw is that actually it was such a mess. There were a lot of, there were thousands of Excel spreadsheets everywhere, but it was very difficult to actually figure out what those spreadsheets, where did they come from and things like this. And the entire thing was just essentially unanalyzable, and they were, because they were not they were difficult to analyze. We also realized that, you know, there were some problem with experimental design to begin with. And so when you look at the time that we spend doing this between the supplies and paying people and the number of people, I mean, that's probably a $500,000 mistake right there. The other thing is that never take like the reproducibility of so the data that I showed is that, you know, essentially we redid the entire experiment once. It took us 6 months the second time we did it, so it was much faster, and because it was better managed, we actually could analyze the data. So the other thing is that it really showed me like that the reproducibility of an experiment should not be taken for granted. Like things that were supposed to be giving the same phenotypes didn't, and it's not because we did any kind of mistake in the experimental design. It was just some of the limitations of the biological assays that we had to work with, and that was the best assays that we could get. So, you know, this is really something that, you know, is very much in the back of my mind moving forward, because when you open the possibilities that things are not as reproducible as you thought they would be, it really changes the way you run your experiments, you analyze your data, and things like this. On the modeling side, well, it's kind of obvious, but, you know, the model was making some some prediction of phenotypes, but the data that we were collecting were not like, you know, one-on-one comparison with, you know, one-on-one. They were not aligned exactly with the mutant, you know, with the model predictions. There was a lot of data reduction that needed to be done in order to go from images of 384 plates with, you know, colonies growing on them into something that would be a phenotype, you know, that would be like a reduced data corresponding to a prediction of the model. And that data reduction process is really part of the modeling. It's really important because different like data reduction pipelines can give you very different phenotypes or, you know, reference to compare your prediction to. And then probably that's one of the most interesting conclusion of this is actually the modeling process is too slow. And, you know, generally we have this assumption that experiments are slower than modeling. But in that case, the model had grown so big and it was such a manual and uncrafted model that when we started, you know, throwing at them like that amount of data, they just couldn't, you know, analyze it anymore. And at some level, it's a little bit of a victory, but it also shows that there is really, you know, there is really opportunities to streamline the modeling process, which raises the issue of, you know, what kind of modeling framework are actually suitable to be streamlined. You know, do you use like black box models that can be really scale up really quickly? Or do you try to build models that are, you know, driven by the biology, but maybe a little bit more difficult to automate? Okay. So at some level, that doesn't look like a very successful project, or, you know, it's actually a project with mixed results. But I think it's interesting, and I'm showing that with some level of confidence, because I'm not the only one struggling with those kind of issues. I mean, there's been quite a bit of literature published results showing that, you know, our community is dealing with major reproducibility issues. You know, there was this paper in 2015 showing that about half of the preclinical research is leading to results that are not reproducible. So that's a major waste of resources. There is all the cell line authentication crisis that, you know, has been, you know, that's part of the reproducibility, I guess. And, you know, when Nature did a survey of how scientists feel about the reproducibility of other people's research or even their own research, the results were, I mean, pretty troubling, because everybody sort of has experienced in one way or another the fact that they were struggling to reproduce either their results or the results of their collaborators. So the other thing that is interesting to look at is really the productivity of life science research. And Deloitte every year publishes a report showing the return of investment of in pharma research that you can use as a proxy for productivity. The sense that, you know, if you're like super productive, then the return on your investment should be very high. If you're not super productive, then the return on investment is going to be low. So this curve is, I've been monitoring this curve for like the last 5 years, and something very interesting is happening. The curve was really like, there was a fairly steady decrease of return on investment up to 2019. And 2020, you know, started seeing a reversal of that trend. COVID probably contributed to the fact that, you know, COVID has been such a major impact on the industry that it certainly has contributed to turning things around, because, you know, a lot of money has been made, and that single project, the vaccine, has been particularly successful. But this doesn't explain everything. And I think that, you know, what the report says is that since 2020, pharma companies have done a much better job transforming, you know, using software and managing their data than they had in the past. So, so synthetic biologists have been talking for 20 years about you know, design, build, learn, test and learn cycle, and you know, every conference you go to, you have, you know, some flavor of this, and everybody's talking about it as if, you know, it was natural and it was working very well and things like this, and they take that for granted. My experience, and I think I'm not the only one, is that this cycle is actually very broken. So you have solutions for design, you have solutions for build, you have some solutions for testing. Putting it all back together and connecting all those different steps is incredibly difficult. And some companies have done a fantastic job, you know, aligning their software and experimental workflows, and, you know, Ginkgo's, Emergen would be some of them. But for, and the biofoundries are trying to do the same kind of, you know, approach on the academic or nonprofit side. But this is very much something that everybody is struggling with. So 2000, so in the last couple of years have been a little bit challenging for my lab. For a couple of reasons. One, money-wise, I was very much struggling to get the yeast work that was really based on my collaboration with people at Virginia Tech. I really struggled to get this funded to continue this line of work. I mean, pretty much every grant that I submitted got turned down. So the lab was sort of running a little bit out of money. But the other thing is that, you know, we got shut down for a number of months, and the people who were working in the lab didn't deal with COVID very well. And so I had all sorts of people on sick leave. I had people with mental issues and things like that, mental health issues. And at the end of 2020, there was nobody left in the lab. I mean, it was just me and myself. Not a single student was left. Not a single, you know, postdoc. Everybody had left. So that was a little bit challenging. And I decided to take advantage of this opportunity to essentially reimagine the lab by changing both the way I was managing the lab, but also changing the topics I was working on. So essentially, I made the decision not to work on yeast anymore. I also, since there was nobody around, I would not embarrass myself by getting back in the lab. So I went back in the lab and tried to figure out where things were and start, running experiments myself, and decided that there was not a single thing in the lab that I could not do. And I've started, rebuilding processes and things like this to work on something that would be more relevant to the NIH. So, and more relevant, more connected to what I was teaching. So I'm teaching, biomanufacturing at CSU. So I decided to work on viral vectors. Viral vectors are viruses that have been engineered for various kind of biomedical applications. So they're used for producing recombinant proteins. Some of them, like adenovirus or VSV, have been used as a chassis to develop vaccines. They're also used in gene therapy and cell therapy as therapeutic agents for gene therapy or cell therapy programs. So it's kind of it's fairly hot in terms of biomanufacturing, and they're an essential component of the development of the next generation of therapies. They have major limitations. The size of the gene that you can insert in those vectors is relatively small. Some of them integrate in the genome, but also there is a critical shortage of manufacturing capability. So the companies that can actually make viral vectors are completely different than the one that make antibodies. It's a very niche market. There is like years of backlog in terms of ability to secure, production runs. There are a number of viruses that are used in this. AAV is one, adenovirus, lentiviral vectors is another one, and VSV is also a virus that's been used both as an anti-cancer candidate. They have a propensity, they tend to infect replicating cells, but it's also been used as a vector to develop the Ebola vaccine. So there are different ones, and from a synthetic biology perspective, it's interesting because it's small enough that you can actually engineer an entire virus without an entire genome without spending a whole lot of money. You can make a lot of variation because most of those genomes are fairly small. So specifically, we're working on a family of vectors that are self-amplifying. And a typical way of working with viral vectors is that you have some kind of plasmid or genetic system that you transfect in cells that leads to the production of viruses in cell culture. You purify the viruses and you inject the viruses in the patient. But we are going to try to change that. The fact that you have to grow the virus in cell culture, and purify them, that's a big part of the bottleneck. So we said, well, what if we could design a DNA molecule that if we inject the DNA molecule into the patient, then the patient starts making the virus. It's much easier to make DNA than to make viruses. But one of the limitations is DNA is not, you can do transfections in cell cultures, but that tends to be fairly inefficient. When you look at the amount of DNA that you need to inject in order to produce, express a gene. And the reason is that because, transcription takes place in the nucleus. So if you inject like 1,000 DNA molecule in a cell through a transfection, you're going to get only a few that are going to get transcribed in the nucleus. And so there is a huge inefficiency in transfection because of nuclear transcription. But there is some evidence that there is also some transcription, and those labels are actually not in the right place. There's some evidence that there is some level of transcription. It's a very small amount of transcription taking place in the cytoplasm. So we're designing vectors that could potentially leverage that and amplify it. And that could be used in a number of different applications. And that would be easier to manufacture than viral vectors. That would be stable at room temperature. That would also be much easier to manufacture than messenger RNA. The other thing that I find interesting in this project is the security implication of the project, because the idea of designing a DNA molecule that could, potentially be injected in a patient with, and produce an infectious agent is kind of interesting from a security perspective. And so, I mean, it's been disclosed. I'm working with, the IBC. I'm not going rogue or anything, but I think that, this is something relatively new and interesting from a policy and it has a number of implications in terms of potential dual use of that technology. So we are very much in the process of like rebuilding that entire design, build, test, and learn cycle. Design and build, it's relatively standard. The testing is going to be, initially cell culture, but eventually we want to get into, in vivo testing. The building part is, fairly standard. That's the kind of thing that a lot of foundries are doing. On the testing side, cell cultures are a little bit more difficult to work with. Mammalian cell cultures are slow, they're expensive, they're sensitive, and so we're still trying to figure out what is the good compromise in terms of how much information we're getting. How do we phenotype our cell culture? Is a plate reader enough? Should we go with flow cytometry or light cell imaging and that kind of things? And we know that at some point we're going to be testing animals, but we have not started thinking about that yet. And in terms of the modeling, we're looking both at mechanistic models that represent molecular interaction in a little bit in the same way as what we were doing in the cell cycle, but also more like generic black box models that are used by data scientists to model pretty much anything. And there is a pros and cons of each approach. The idea is that we are going to be generating a lot of variants of those viruses, so and look at endpoints phenotypes or a few data points per variance, rather than do a very deep phenotyping of a smaller number of variants. And the design objective is to control the virulence of those viruses. So in the sense that, we want to be able to reduce the expression, but without increasing the variability and the stochasticity of their behavior, which is a little bit of a catch-22. So I don't have anything to show quite yet or on this, but this is coming. But this is giving me an opportunity to actually think about how do we manage the research data. And full disclosure, I'm going to be talking about Genofab. So this is a company, so I have a conflict of interest and blah, blah, blah. But I'm going to try not to present that as a sales speech, but more as something scientifically interesting. So the first question is, what kind of data do we want to manage? And it's fairly common, when you're using a laboratory information management systems, people want to manage their, supplies and DNA solutions, cultures, I mean, any kind of physical samples. There is another kind of record that needs to be managed is the data set. So like, for instance, if you're running a sequencing experiment, you need, your data is in files that have your sequencing reads. If, any kind of instrument that you are using is going to be producing files. And it's very common that those files are actually managed in a completely different way in a completely different system than your samples. So even if you have like a good inventory of your samples, it's not unusual because, you know, those files are big that they sit in some kind of shared server, and before you know it, you are, you know, like very quickly you can end up in a situation where you have a ton of files that cost you a lot of money to produce, and you are unable to actually track down how those files relate to the experiment that you have run. And that, you know, because there's a disconnect between the two. Another part that is also important is keeping track of the computational steps, or, you know, things like primer design, sequencing read assemblies, any kind of data reduction workflow. All of this is things that have parameters that need to be connected to your experimental data, your sample, and things like this. Otherwise, you know, that may end up being the least reproducible part of your workflow. Supply chain is an important component as well. Like, you know, you need to keep track of your orders, and not from an accounting perspective, but you need, you know, you need to be able to connect like an oligo order or gene synthesis order to the rest of your workflow, because the output of that operation is going to go into, you know, things that you're doing in your lab. And in addition to that, you know, it can be useful to keep track of things like compliance records, training records, equipment maintenance, and all of things that, you know, you need to keep track of to manage a lab. So the way we are representing the data is really using 3 layers of abstraction. One is recognizing that, so we call the catalog essentially the database where we put all those data. And one is to define a type which corresponds to a class of objects with the same data structure. So things like equipment or supplies or, you know, a cell culture can all be represented, you know, each with a different data model. You cannot represent a cell culture and an equipment with the same kind of data. You need to record different kind of data. So once you have defined that data structure for a particular class of objects, then you are going to have individual objects in this, and that's going to be a member. So, you know, if you have supplies, for instance, your T75 flask or your DMEM are going to be example of supplies. So they are like catalog entries. But now each of those entries are going to have a number of instances. So if I say that my lab is going to be using like, you know, T75 flask, I'm going to place a number of orders. If I'm using a certain media preparation, I'm going to make that media preparation multiple times. So, you know, so that's why we have like 3 categories from like classes of objects with a common data model all the way to physical instance, which are essentially replicates of something. So the way we define those classes in GenoFab is, you know, there's a graphical editor where you can you know, edit or define a catalog type, and you have 2 lists of data fields. You have the entry fields, which are common to all the physical instances, and then you have the fields corresponding to the different replicates. So another way to look at this is that the data associated with the entries, that's more or less a parameter of an experiment. Experiment being defined in a very broad way, you know, like placing an order for a supply could be seen as an experiment at some level. But it can be applied to a cell culture, it can be applied to a DNA extraction or a sequencing operation and things like this. So these are the things that you're going to be controlling. And the data that you're getting, that you're associating with the physical instances are data that you don't control, that you observe. And the different instances are essentially replicates. So another analogy is, you know, when you go to the grocery store, you have a shopping list, and your shopping list is going to be more or less the same every week. It may include, okay, I need to get like 2% milk. So that's a description of an object that you're going to get. But the content of your shopping cart includes a physical object, which includes, you know, data like the expiration date that you don't control. So a big challenge of ensuring the reproducibility of what you're doing in the lab is to actually keep track of what you have in the lab. And that's where labels are so important. So the software includes like a way of printing barcoded labels that are uniquely identify everything that you have in your inventory. And that is incredibly useful because it avoids the problem of having to give names to your samples, which can be very tedious after a while or very confusing. It also makes it possible to, very much streamline, make sure that everybody in the lab can clearly identify like a supply, a sample, and things like this. It removes a lot of the ambiguity that is associated with poor labeling. There are some questions on the chat. I cannot give the presentation and answer the questions on the chat. So, can we have the conversation? Either if you want to have the question, ask the question, or keep it for the end. Okay. And so we started analyzing some of the data that we produce with this, and just looking at the reproducibility and sources of variation of a couple experiments that we have run on different sequences with enough numbers. So one that I'm presenting here is like plasmid mini prep. So we did 3 replicas of 3 different plasmids that were very close to one another. It's all the same backbone. It's pretty much the same size. There's no major differences. And we applied some, statistics on that. And what we saw is that there was very significant differences in the yield of the DNA extraction across plasmids. And that is a little bit strange because we don't think that there is anything really different between those plasmids. So it's not like, one is a high copy number and they're all in the same bacterial strain and things like this. But there was no significant differences of yield across repetition of the purification. However, we could see that there was some statistically significant differences of purity, at least based on A260 over A280. But the differences are small enough across repetition, but the differences are small enough that it really doesn't have any biological influence. So it's just one example of the kind of analysis that you can do when you properly manage your data. So the other thing that is important is, there is a flow of data in the lab. So, it all starts with supplies that you're ordering, and then you mix the supplies, you make solutions, and you use those solutions to make media, and then you do cell culture. So, things are going from things that you're buying and all the way to the data that you produce. So there is this notion of transformation. And so we have, when we define those, data models, we can include links showing how records are going to be connected to things that were defined previously. So in this case, I'm saying, that this particular cell culture media is connected to a particular protocol. Sorry, it's going to be it's going to be connected to some supplies that I'm buying and some aliquots of another supply. And when I'm defining this at the entry level, now what I can do is actually keep track at a particular, I can associate a particular version, particular preparation of media with a particular version of my protocol, a particular order of the media, and a particular aliquot of the additive. And so when you look at this like graphically, now you have different classes that communicate to each other. So you can establish links between different classes of objects, and under those links, so it's a little bit like a road, and then under those links you have like individual cases or individual items that are connected with one another, so that you can go through a cell culture and be able to track down the particular order. You have complete traceability in terms of the order of supplies that you've used, the particular order of supplies that you've used, and things like this. Okay, so this is, we call this model like a data flow, and I think it's very different from a workflow. A number of systems have workflows where you have a sequence of tasks that are related to one another. And they tend to be fairly deterministic and linear. And the kind of data model that we are proposing here, it's much more like a grid of connections. It's like roads, and they are used by many cars, but they are not always, cars can go in different directions. It's not as deterministic. They have alternative routes. It's much more flexible because a grid of connections, can change automatically by adding new nodes or new relation between existing nodes. And we started developing like functional modules for different aspects, whether it's DNA vector constructions, bacterial cultures, cell cultures, and so on. So I'm fairly confident that this approach is going to be able to build those links between the different aspects of the design, build, test, and learn cycle. I'm very confident that this is going to work fairly well, but it's still a work in progress because we need to demonstrate that on a couple of projects. So in terms of conclusions, a few take-home messages. A lot of research projects look like this. You assign a project to a grad student or a postdoc, and they just try to wing their way through, a publication. And they try to figure out a way of, putting the experiments, publishing the data, and that kind of things. And that raises, it's not terribly efficient because there is a lot of reinventing the hot water that takes place in this, and it makes also reproducibility difficult. So I think that it's more, as PIs, or we need to be thinking about what, how do we build a road as opposed to clearing the bush just to find our way through the thing. And it requires a little bit of an investment, but it's much more reusable. It's going to give you more data. It's going to be cheaper in the long run. The other thing is that you need to make sure that those roads connect to one another. And every kit that we buy comes with a little manual, but there's a lot of protocol engineering to customize those protocols or develop SOPs that that gracefully live with one another, so that that you can go from one step to the process to the other one. Otherwise, it leaves way too much interpretation, and that's anything can happen. The last point is that it's essential to characterize the variation, the sources of variation of your processors. Replicates are often seen as a demonstration that your process is stable, but replicating your process is actually a way of stabilizing your process. That's a way to give a process that is going to be less variable, and the sources of variations are going to be understood, because you can end up in a situation where there's so much variability in your process that really the thing that is determining what the data that you observe is not necessarily the thing that you have designed. It may be just like anything in between your design and your data. So stable processes take a little bit of time to put in place, but they save time, they save money, and they give you publishable data. And at the end of the day, I think that what we are learning is that really research used to be something that we would do on our own, and everybody, every grad student would have his little project and that kind of things. But as the size of the research enterprise is increasing, it's becoming really something that there's a division of labor, and it's really much more like a teamwork, where really the role of the scientist is no longer really to produce the data, is to think about what kind of data needs to be collected and interpreting the data that are getting there. And that's a little bit like the composer of a piece of music. The lab manager is probably the most underappreciated contributor to a science project, because he's the one who is really going to, their job is really to make sure that the data that are being produced are actually stable, are usable, and that kind of thing. So he's really the one coordinating data production effort. And you can do that without necessarily understanding the goal of the research project. And the data production is, getting more and more like a factory job. And that's something that, if your lab is properly managed and your data is properly organized, you can train someone very quickly to produce quality data without having to have them, to have 10 years of postdoctoral experience to understand the research project that those that are going to support. So number of collaborators on the cell cycle, self-amplifying vector, that's a smaller team, some former collaborators and some of the people at Genofab. If you want to contact me, you can reach me on my website. If you want to learn more about the software, book a demo on genofab.com. We have actually a program for academics, so I'll be happy to talk to you about that. And I'd be happy to take any question you may have.
Thank you so much. Jean, there's a lot of questions in chat.
Yeah.
If you could read the question before you answer it, that's going to help with people that watch the recording.
Okay. What's the most complex system you could model with this model? Would it be possible reproduction of? What model are we talking about here?
I think that question showed up in the first half of your talk.
Yeah, so I don't know if it's about the cell cycle model. I think that possibly just makes, it would make the model more complicated. And the cell cycle model that we showed that a number, I didn't develop it. I mean, it was something that was developed by John Tyson. I know that a number of people in the community were a little bit troubled by the size of the model. And, there was a lot of criticism because it was just so big. So, sometimes modeling, it's about simplifying things. So I don't know what to answer to that question. Okay, so I think I have a few messages of sympathy. Thank you very much. Could you please comment on biocontainment and safety of viral-based vaccines? Is that a solved problem? So what is the question here? Are the viruses going to propagate, and is that the question? If you're viral-based vaccines, so I guess if the question is, is there a biosafety risk with viral vectored vaccines? Possibly, that's why there are clinical trials. I think that the vaccines that are, like for instance, in the case of Ebola, the Ebola vaccine, which is the one that, I know fairly well, VSV is a virus that doesn't make, it makes cattle pretty sick, but it makes, it just gives you mild cold symptoms. So it's a virus that is not particularly aggressive to humans. And when you just put an antigen on top of it, it just reduces their fitness. So I think that whenever you're dealing with a live vaccine, the question is, you need to attenuate it enough that the patient is going to recover from them and it's not going to propagate in the population. The immune system is going to be able to control the infection. But you don't want it to be so attenuated that it just doesn't elicit the immune. So there's quite a bit of fine-tuning that needs to take place there. So there's a question about the integration of all the computational tools, I guess. So it's very much, we're using a lot of software tools during our workflows, to implement our workflows. I think it's really important right now. Those tools are all, separate from one another. And I think that if we want to, make the computational pipelines more reproducible, they need to be integrated into the entire workflow. So that, like, for instance, just like a sequencing experiment, you go from a DNA, from a library to a set of sequencing reads. The next step in the process should be to have not a bioinformatician who is going to be developing some kind of custom script to analyze your data, but a sequence assembly pipeline that is going to be robust enough that you can tweak the parameters and you go from your assembly, because you can do. So all those tools need to be connected to your data management system. That's very much. We have a couple in the works at this point, but that's I think that's absolutely critical, and I don't think that a lot of people have a good solution to that. Even the Agile Biofoundry doesn't seem to have. I talked to them like several times, and it seems to be a little bit still all over the place.
So actually, can I add one more question to clarify my question?
Yes.
So yeah, that's what wonderful answer for this kind of challenging question. So what I envision is basically, the student. I love your last slide about the role of each different person. I mean, the composer or whatever. And what I envision was basically, student and postdoc and even professor just only create new idea, new vision, and then new kind of concept or new kind of experimental design. And then once you did, basically the rest of the work is done by robot and so on, and even not necessarily require the technician. That's kind of how I think. I mean, not tomorrow, but probably 20 years later and so on. But I believe to do that, there are much more kind of challenging problems we need to solve. And one of them you mentioned integration, because my student some of them using AlphaFold and then to do the protein engineering, and then the other network model and so on. But everything different place, not the integrated together. So, but on top of the integration, there would be some other challenges. I mean, to do only we focus on those design and then all the cloning done by machine and then those kind of things. And then just how we could do that by using your fascinating models and then integrating everything together. So that was actually the question I wanted to ask. So I mean, anyway, a brilliant talk. I love it.
Okay. So there was a question about the data management software for collaboration. So this is very much on our product development roadmap. We think that it's absolutely critical that we have an easier way of, giving different stakeholders the data they need to do their share, but it doesn't mean sharing everything. And whether it's with a vendor, being able to streamline, like, for instance, the synthetic gene order would be very useful, because right now there's a lot of friction in this, but it could also be, going through different collaborations. So it's very much on our roadmap. It's not something that is implemented at this point, but this is a pretty high priority. Recommendation for an already established lab. That's a difficult question. So I have a lot of answers to that, and I would suggest that you schedule it has to do a lot with how the PI wants to manage a lab. So if there is if you feel a need that everybody managing their thing on their own is not the right way to go, and it's not the right way to go because it's very inefficient, it wastes a lot of resources and things like this. a good way is really starting with the easiest one, is managing your inventory and then managing your, your equipment and progressively building, building on top of that. And sort of separating also the notebook part, which is really note taking, versus the management of the data, which is a slightly different layer. The notebook is not the place. So the notebook is not the place, is not the place to manage data, because any data that you put in a lab notebook is essentially, it's a data graveyard. There is no way you can analyze data when it's embedded in a wiki kind of thing. Okay, a notebook is really meant to be read by humans, not read by analytic software. So there is a need to separate the note taking that goes with running an experiment versus a collection of data that you have well established. So that would be a very short answer. I'd be happy to follow up with you if you send me an email. So the negative result question, I don't have a good answer to that. Some of you know that I'm an editor of a journal. So we've been managing a paper that produces some negative results, and some of you are co-authors of that paper. And the really difficult part is that a negative result is not necessarily a result. There's always, it's easy for the reviewers to say, well, there's this other thing and that kind of thing. So I understand the need, but it's a tricky issue. I think that what an alternative to that is, instead of seeing it in a binary, whether it's positive or negative, trying to explore statistically what are the factors that are affecting the variability of your data is something that is useful, because that you can say, well, okay, I'm doing this, and when I'm doing this, I'm exploring how different aspects of my process leads to the data that I am control the variability of the data that I'm producing. And that sometimes that you can come up with results that are statistically sound, even though you don't completely understand them. And that that may be like the alternative, the middle ground between the negative results and the positive results, where you say, okay, there are some things that I can clearly identify as affecting my data, and I don't really know why. So the role of professors, that's a tricky one. Yeah, I was thinking about that. So, I mean, you would like to think that the professors are actually the one doing the research, but in many cases, professors are just like writing the pitch, getting the money, and for the most part, like students and postdocs are the one really doing the research. I mean, and we are paying them and we are helping them, we are mentoring them, but so, sometimes I feel that that actually the role of the PI is really to be the lab manager. That this is really something that, because the PI is essentially, responsible to make sure that the people working with them can produce quality data, and that you can produce all the data that you need in a scalable way. So, at the end of the day, it's a little bit sad, because a lot of us don't really have the resources to pay for a lab manager, or a good lab manager. And so at the end of the day, I'm rebuilding my lab, and I'm trying to figure out, what role I give to the people that I'm hiring. The one that I'm the most reluctant to give up is actually the lab management part, because experience proves that it's actually very difficult to find a lab manager who understands how the management of the lab relates to the research program. And okay, I think that's the end of the questions.
I think you went through them all. Thank you so much. I was particularly intrigued, and I'm glad someone asked the biosafety question, because I was intrigued about that too, and that's always creeped me out a little bit.
Yeah.
Yeah, thank you so much.
Thank you so much for the invitation. I really enjoy it.
Thank you for giving a talk, and thanks for answering all the questions. And thanks everyone else. And
Have a
see
good
you
week.
next week. You too. Have a great week.
Bye-bye.
Bye.
Our back tensor job.
Yeah, thanks. Bye.
Thank you so much. Wonderful talk. I love it.
Bye.
Thanks. Bye.
Bye.